I know. Okay. So this is why I was kind of making a face earlier, because I've made a fresh culture specifically to do at least a demo of this, but I don't think we're going to have time to get to it today, which kind of bums me out because then I have to make another fresh culture next week, but hell, I can do it. So we're going to look at our motility stabs. That's the motility test. We're going to talk about how it is they're motile by flagella and the different arrangements that your bacteria could have. And we're going to talk about the Thioglycollate, the FDM tubes. As I said, I'm going to take yours out of the refrigerator after I lecture so you can record your results, and I'll help you with that. And then we're going to talk about the anaerobic jar. We no longer do this. My jar is down here. These are apparently $500 a piece. Yeah, this plastic jar is $500. We don't have enough of them currently that the seal isn't defective on them. So we don't do this experiment anymore. We used to do it in the past. We don't have enough for each class to do it. So I have pictures and we're going to talk about it. But I guess I need to write another grant. Right? Maybe this will be my next grant. Let's get us some new anaerobic jars. Or some new lids, I guess, or seals. Yeah, this is the jar.
You envision the Mason jar?
Yes,
Me too.
me too.
Well, I have one of those too, right? So a jar like this, you could put a candle in it and do a candle jar, right? The problem with this jar, though, even though it's big, this lid isn't big enough. This opening's not big enough for me to get a petri dish in here. Otherwise, we would use this. But we just had this as a demo. But yeah, like, so this one, see these rubber seals? They're not holding. We need like new rubber or something. Yeah, you would think. Yeah. So that one's actually a good one. The bad ones are further back. The problem is I don't have enough for each class. So we're going to talk about motility today. Some of the terms that you need to know, which are covered in that other chapter, and I didn't add them to your homework packet, but you want to make sure that you write them down, right, or print out this PowerPoint or write it down from the PowerPoint. Those of you guys in lecture probably already know these, right? We went over this, although it was a while ago. So mono means how many flagella? One. Trichus refers to the flagella because it looks like a hair-like appendage. Trichus actually means hair in some language. Sometimes you'll also see the term polar because, as you'll see when we look at the pictures, it extends from one end, right, to one pole of the organism. So ones that are monotrichous or polar tend to have one single flagella at one end of the organism. Which, if you have a cocci, is there an end? No. There's very few cocci, though, that actually have flagella. Amphitrichous, amphi meaning opposite, so there's one at either end, right? So at opposite ends, there is a flagella. Lopho refers to a clustering. So for this one, it's a clustering of them usually at one end or at both ends. Right? The term really just refers to multiple flagella at an end or sometimes at both ends. So as I said, we'll look at some pictures for these as well. Peritrichous, which normally I would tell you guys which ones are peritrichous, but I want you to find that out if you want to earn bonus points this time about your organism. So peritrichous are completely covered. The whole surface is covered in flagella, multiple flagella. So it's like scary cousin it. So based on that definition, right, first of all, what shape is this bacteria? It's bacillus or rod-shaped, right? A lot of times we say rod instead of bacillus because bacillus is also a genus name, so people get confused, right? So he's covered in flagella, right? Scary cousin it. So what term? Peritrichus. Right? This one, again, what shape? Rod or bacillus, right? One single flagella at one end. Monotrichus or polar. What shape is this? It's not a spirochete, spiralium, right? And a lot of these guys are motile by flagella, where spirochetes, remember, have internal flagella known as axial filaments, and they're more helical and they corkscrew through their environment. We don't deal with any of those in this lab, right? They're pathogenic. Things like, Lyme disease, ain't messing with it. We don't even have any of these guys, right? You'll notice on your report you have two choices, cocci or bacillus. So exciting. But it is, kind of, right? So a lot of these guys have external flagella. And so this one has multiple flagella. Do you see at this end and this end? So the term for this one would be Lophotrichus. And this one has one flagella at either end. Again, another spiralium. Amphitrichus. Yeah. And so these are probably electron micrographs where they use electron microscopes. These are light microscope probably pictures using a special stain called the flagella stain, where they actually make the flagella larger than they actually are so you can see them within the limit of resolution of our microscope. Because remember, we're at 200 nanometers, that's it. This is really small. And most flagella are smaller than that. So you're not going to see them unless we stain them, and this stain actually adds thickness. So most women understand this analogy. It's like adding mascara, right, to help visualize the flagella. I've tried with my summer STEM students to do the flagella stain, and we have unfortunately not been successful. It is not apparently an easy stain to do. It's one of the reasons why we don't do it in this lab. So for your organisms, you're going to know it's motile, and I can tell you the reason it's motile is because it has flagella. You would, once you find out what your organism is, would have to research to find out how many flagella and how they're arranged. And you could do that for bonus points for your poster. Okay, so I wanted to do a hang drop preparation today of one of the ones that a lot of times it looks negative in your guys' tubes, but it's actually motile. And that's because there are some limitations to these tests. And that's why sometimes it's better to do 2 tests as opposed to just 1. And then for you guys, right, you did a whole bunch of stains, but we're doing a whole bunch of tests in those tubes and even more tests we're going to do later. So we're going to gather all that data, and you'll notice in the last page of your packet is all the known data for all the known organisms we've worked with in this lab. So once you gather all your data, you can compare it to the chart and by process of elimination figure out what your organism is. Hopefully, right? You get all your stuff right. So we could put some water on a slide, right, and add a cover slip and do just a simple wet mount. One of the reasons why I don't like to do that one is it's not as easy to focus and visualize than the hang drop. The hang drop is a little bit more technically challenging, which is usually why I help students, or I at least do a demo for you guys so you can see it. You need a depression well slide, which are expensive slides that I keep stashed in the instructor's desk for one. And so that you can have the drop of water containing the bacteria hang sometimes right in there in that depression. Really hard to focus. And then there's an additional problem, is we don't actually stain them. So, how? Exactly, because that's why we stain them most of the time, right, is so we can see them. So how are we going to see them? So we have to kind of decrease the light to help us visualize them. Yeah, so it makes it a whole lot harder to see them. But I can tell you, it can be done. It's not an easy technique, but it can be done. So I have some videos, some YouTube videos and stuff of ones I've done in the past. And again, I think I'm going to save this one to next time because I wasn't able to set it up. I should have set it up while Peter was talking, but I was mesmerized by him too. So, all right. So, and today, just like, it's just one thing after another. Those darn surveys. They told us yesterday, that we were doing them this semester. Yeah, I don't know. Yeah, I'm not going to bitch. Okay, that's life. So what are some complications? So if you were doing a wet mount or a hang drop slide, what are some problems that you would run into? So you would use controls, just like we have positive and negative controls for gram stain. You would use controls to help you, especially with difficult techniques like this. So if you had an organism you knew was motile, right? And you put it on the slide, but you put so many of them, they're all crowded in. Would that stop them from moving? Yeah, think about being in a crowded space, right? It restricts movement, and so it makes it much harder to see. So the problem with that is if you have a motile but you put too many of them on your slide, right, or in that wet mount, you may not see them move, right? So you write down negative, right? On your paper, you're like, oh yeah, mine's non-motile, negative, moving on. Then you go to try and figure out which organism you have, and you're like, they're not matching up. Why aren't they matching up? Well, because you didn't recognize that you put too many on the slide, right? And so that wasn't a true negative result. That's what we call a false negative result. And so that's something you have to look out for, right? These type of complications. You don't want to overcrowd, because if it's a motile, you're going to crowd them in and you're not going to see them move. And you're going to end up with a false negative result. Would this make a non-motile move? Right? If you crowd them in, no big deal. They weren't going to move anyways, right? So this wouldn't have any effect on a non-motile, but you have to worry about this complication for your motiles, right? Because it's going to give you false results, specifically a false negative. What happens if they attach to the slide? Yeah, they can't move as well. And actually, there's a cool video out there on YouTube where one of them is spinning like this because he's stuck to the slide. It's kind of cool. But, yeah, if they really attach to a large degree to that slide, then that's going to stop them from moving, and you're going to end up with another, what type of result? Another false negative result. Will this make non-motiles move? No. So again, this is not a problem we worry about for motiles. The good news is microscopic organisms, right? If you don't overcrowd it, you're still probably going to have plenty to look at. Hopefully somebody didn't attach and is moving. There's no real way to necessarily avoid this, right? But what you can do is scan a large surface area on your slide, right? And make sure that you look at lots of cells as opposed to just a few. Does that make sense? Okay. So a receding waterline is an interesting one, and it's definitely a complication you tend to get more with a wet mount as opposed to a hang drop, which is another reason why I prefer the hang drop over the wet mount preparation. So this is where the water is moving. So think about this. Anything that's in water also moves, right? Like when loggers cut logs and they used to throw them in the river to send them downriver, they would float downriver in the direction of the water. Well, think about this. Bacteria, we just stuck them in water, right? Even if they're non-motile, if the water's moving, they are going to move. But how do you spot this? So we can't really necessarily stop this. But how do we know that this is happening and the cells aren't moving? Exactly. They're all going in the same direction, and although bacteria can move pretty darn fast, they really move. Water moves faster most of the time, right, in a receding waterline. So it's all in one direction. It's probably a receding waterline. Wait for it to settle, right? If it looks like it's going really fast and all in the same direction, you're probably dealing with a receding waterline. So is that a problem for motiles, though? So if you see it moving and you write positive and it was positive, no big deal, right? That problem comes in when you've got non-motiles, right? Because you see movement, don't you? So you write down positive, but oops, you didn't recognize it wasn't bacterial movement, it was water. So this would give you a false positive result. What would be the problem with using an old culture? So if your culture is old, are there more alive or dead cells? Dead. Do dead cells move? No. Remember, this is not zombie class, right? No zombie bacteria, right? Dead bacteria don't move. So if you have mostly dead ones, right, in a motile culture, are they going to move? No, and so you're going to end up with another false negative. Being dead, is that going to make them move? No, so it's not a problem for our non-motals, right? No zombies. It's a zombie-free zone. Does that make sense to you guys? Right? So when we know about the limitations and the complications, right, we can do the best we can to avoid or recognize these situations to make sure we don't get false results. Right? So we really want to look out for these conditions of false negatives for our motals, and that one condition that can end up with a false positive of moving our non-motals on us, of that receding water line. So as I said, I've got some cool YouTubes. There's some links here, but I'm not going to play them right now, and take a look at them before next week, and then I'm going to set up a live demo for us for next week. So what is Brownian motion, though? Yeah, water molecules are constantly colliding with each other and bouncing off of each other. Anything else exposed to that water also can get bumped into, right, and moved. And so this creates a vibration effect. And there's one stain that we used to do that was kind of an aqueous stain in one of the labs I worked in, was an India ink stain for capsule staining. And I'm really glad we use a different technique here, because that one, you'd end up with little droplets of black dye, and it was more of a liquid type situation, and they would vibrate, right? So if you looked at it for a long period of time, right, and they're vibrating like this because of Brownian motion, because they're colliding into each other, it would give me a headache, right, looking at this vibration. So there's some videos of that as well. And this vibration is due to thermal energy, right? So heat energy. So what on our microscope kind of makes a problem for us? Oh, I'm skipping ahead. Sorry. Before that. So why don't we stain them? We would kill them. And again, no zombie bacteria, so they won't move. So one of the things that we do is we decrease the light. So what I usually do is I close down the iris diaphragm, right, underneath the condenser. We close it down just like the iris in your eye, and we decrease the amount of light going through. This less light helps us. It's enough to help distinguish those relatively transparent cells from the liquid they're in. So you can see the shadows. So it's very much like, you know, a room with a night light, right? You see a lot more shadows than obviously a pitch dark room. You're not going to see anything. Or a very bright situation, right? The first thing you do when you give a kid a flashlight usually is, what do they do to you? Don't they shine it right in your eyes? I mean, like, really, you're like, oh, dude. And that's just too much light, right? It's just like most of the time for you guys, because you're not used to it, when you have the full light on, it just blinds you, right? Because it's just too much light. What you need to do on your microscope, just as your eyes will do eventually, is close down your iris diaphragm, right, on your microscope. Let less light in. And other animals are much better at that because their iris are different, right? You know, notice how cats' eyes have that slit, right? And so they can really close it down, but yet still get pretty good vision. It's pretty amazing. So Brownian motion, as we were talking about, this is related to heat. So what on our microscope heats up and creates a problem for us, and will exacerbate Brownian motion sometimes? The light bulb, right? A lot of the light bulbs, especially the microscopes I like to use, have halogen light bulbs. These get hot. The newer microscopes, though, nowadays use LEDs, which don't get hot, which is nice. I wish we had LEDs. But, again, not a huge deal. So the next is our motility test, which you guys did, where you stabbed into this media. So this media is referred to as being semi-solid, not solid. So although it seemed kind of solid, didn't it? But if you were to touch it, it's not as solid as like our plates or even those slants that we use. Yeah, it shakes like Jell-O, right? So the reason for that, so why isn't it solid? Why don't we want it, you know, really thick? Yeah, they won't be able to move. We want some space between the molecules so that the bacteria can move, because we want to be able to record movement, right? Motility. But because it is kind of suspended, kind of like a Jell-O or gelatinous type situation, we can see the growth in a pattern, right, as it spreads out. So what does a positive test look like? Which one of these tubes is positive? It's actually the one over here that has positive question mark, right? It's really hard to see in this tube because the reagent that we use, TTC, is not in this tube. Okay? And so the bacteria have actually spread out. That's why it's cloudy. If you look at it a little bit more detail, you notice how it's cloudy. Where this one is definitely negative because the bacteria stayed exactly where they stuck it in the tube. You're supposed to look at this first. Then we'll look at those. So for our tubes, but a positive test is much easier to see for some of these tubes because we can see the spreading, right? So for a negative, no matter which way you turn it, it should be a single straight stab line, right? Because the bacteria are just where we put them. But for a positive result, if you spin that around 360 degrees, it should look like a little tornado, where they've spread out, right? They didn't stay where we put them. They have traveled outward. So it almost looks like a little tornado. Now for some of yours, they've moved so much that your whole tube is red, right? Some of you guys, your whole tube is red, and you really don't even see the tornado. But the fact that the whole tube is red tells you it's motile, right? If you actually can see the stab line, and you don't see any spreading from that stab line, then yours is probably negative. Probably negative, right? There's one organism that doesn't spread well, right? And you'll find out when you look at your slides today. Okay. So you can see why we use this reagent, right? This is really beneficial, this TTC, because it is colorless in its soluble form. Soluble meaning that it goes all throughout the media, right? It becomes insoluble, stays put, when the organisms oxidize it. And it also changes color. It changes to red. Oh, excuse me. When they reduce it. Not oxidize it. When they reduce it. When they add electrons to it. It's red. So that's why we use it, is to help visualize where the growth is in the tube. That's why we use this reagent. Because of these properties of being colorless. Although you guys will notice our tubes started out pink, right? So the problem is that when we make these tubes and we add the TTC in ahead of time, in the autoclave, it's getting reduced. And we don't know how to fix that. So next week and this week I'm going to be playing around. And I think what we're going to do in the future is have you guys add that reagent to the tube right before you add your organisms instead of beforehand, because the tubes are coming out too pink and it makes it harder for you guys to see your results.
How would we add the reagent if it's already?
So we're going to have to add it aseptically with a sterile filter so that the solution that we add is sterile. But it'll be good for you guys. Instead of having Paula add it to like 120 tubes, each student can add it to their own tube. Right? So we're going to work out the kinks of that, and I'm hoping to have some new fresh demos. You can see before we had some ones where the tube is pretty clear, right? But as you can see, they're getting pretty dried out. Those are ones I've been stashing in the fridge for a while. But I'm hoping to make some fresh ones that look as pretty as, if not better than these pictures. So why is it important that we insert in and draw it straight back out? Yeah, if you move it around, right? If you were to put it in and then go like this and shake it out, right? You're putting the bacteria all throughout your tube as you're inoculating it, right? So then when you go back and look at it, you're going to be like, oh, it's motile. No, you moved it. It didn't move. You moved it. So this will end up, if you don't recognize the fact that you improperly did the technique, right, then you would think maybe it's motile. And some of them, depending on the age of the media and the organism, we get some weird stabs sometimes. So notice this one, see how it's kind of sticking out? This one is definitely a bad stab, right? The person went in and moved over before they pulled it out. So this one you can recognize though, because this is only on one plane. If you turn this tube sideways, right, it'll look flat. This one, again, I think either they stabbed it twice. Do you see how there's a line here and a line here? They either stabbed it twice or they stabbed in, moved over, pulled out. Again, this one you should be able to recognize if you turn the tube that that's what happened. It was a bad stab. And I've got some examples of bad stabs. So that's false positive, right? If you got a bad stab, right, and you don't recognize, right, that you moved it, it didn't move. What about false negative? Why would you maybe not see movement? Under inoculation, right? Not enough bacteria, so not enough growth that you don't see the spreading. Good. The media is too solid, right? It's too old, right? You put it at the wrong temperature. Excellent. Growth conditions. Good, good. You guys have got your thinking caps on today. I'm impressed. So that's our false positive, right? So here's a nice clear negative, right? This one, right, something's not quite right, right? And you might think that that's actual movement, and it's not. The spread is not uniform. Do you see how that is? It's like a weird pattern to it. Right, that's a negative. So, false positive is poor stabbing technique or misinterpretation of some of the ones that have these weird patterns. The good news is we don't use either one of these organisms anymore because they're pathogens. So most of your results, I think, are not too ambiguous. False negatives, right? So look at this one. See how light that is? Right? It's not very thick line. That was an under-inoculated one. Maybe they didn't put enough bacteria in it or not enough live bacteria in it, right? It might have been an old culture, so there wasn't a lot of bacteria, right? Might be a slow grower, right? May have been grown at the wrong temperature, something like that. So you would repeat that. So false negatives, not enough live viable cells, right? Not a good culture. If the media is too solid, and again, our lab prep person knows how to make it, right? But if it's too old, that could cause the problem because it'll dry out. So we usually get rid of it, right, when it's too old. So why is it important that when it's reduced, it's in that clear state that, when it's reduced in that red state, that it's insoluble? Right. Insoluble means it won't move on its own, right? So when it gets reduced by the organism and turns red, it becomes insoluble. So it stays right next to the organism. Where if it was soluble, it could what? The red could spread on its own. And then we don't know, right? Is that the organism? Right? So it's effective because of the fact that it's insoluble when it turns red. It doesn't move on its own. Where it is soluble, it's all throughout the media, right? So no matter where we stab, when it's colorless. Our problem is, right, some of it's getting reduced before we even stab it. So it's pink. So remember, for each one of these, we could apply the scientific method, right? Why are we doing this? How do we do it? What are our results, and what do these results mean? So for our FTM tubes, we use these to determine the oxygen requirements of your organism. Okay, so these special tubes are set up to be able to detect that. So why is oxygen important? Why is it important? No, we're moving on to another test. So why do we want to know if your organism is using oxygen or can grow in the presence of oxygen? Figure out if it's using oxygen for its metabolism, right? Or if it can survive in the presence of oxygen. Which is true for all your organisms, by the way, because again, we don't have any anaerobic jars, right? We don't have the special equipment. Nobody's working with an anaerobe, y'all. Don't write anaerobe on your report. You will get it wrong. Nobody's working with a strict or obligate anaerobe. It's not possible in this lab. The oxygen would kill it. So oxygen plays some very important roles for energy and growth in cells, right? We use it to accept the electrons at the end of our electron transport chain, right? As we break down sugars for energy. So I'm going to skip over these next couple of slides, right, in the interest of saving time. If you guys don't remember things like the Krebs cycle and stuff like that and you need to review, I'm going to post this section of recording of an older lecture for you guys, okay? If you need a review of this to kind of fill in the gaps about why the testing we're doing is important for your organisms. Which is why I wanted to post it ahead of time. But, so, the bottom line. The important stuff, right? If an organism can grow in a media that contains sugars but lacks compounds necessary for anaerobic respiration, then they have two options. They can grow well via aerobic respiration using oxygen, or they can grow poorly via fermentation, which does not require oxygen. Okay, so some of them have this choice. In reality, not all organisms have that ability to choose, right? Like us, right? We're strict aerobes. What happens when there's no oxygen? We die. There's no other option. But some microorganisms, you can imagine, their environments can be changing as it relates to oxygen concentrations and availability. So it is in their best interest to be able to have choices, right? Because if they don't have choices, then they would die. So life on this planet, actually, they believe, happened without oxygen. And then organisms evolved that could produce oxygen, like the cyanobacteria and the algae in our oceans, actually produce excess oxygen that make life for organisms like ourselves that are aerobic possible. Right? So it wasn't until that happened that we were possible as far as being aerobic, or any other organisms like us that are strictly require the use of oxygen. The other problem is oxygen can be toxic, and it's toxic even for us. So we have protection against the toxic forms of oxygen. And we're going to run a test in a couple of weeks to see if your organism has that enzyme. But considering we're in an oxygen environment, what do you think? If they don't have that enzyme, what would happen to them? They would die. So when we say aerotolerance, we're talking about just that. Can they tolerate oxygen? Right? Do they have protection against it? Do they have enzymes to protect themselves so oxygen won't kill them? Can they tolerate it? Not all of them will use it, but can they tolerate it? So strict or obligate aerobes like ourselves, by definition, have to have oxygen. It is required for our metabolism. We are strictly aerobic respiration. We cannot survive without it. We don't have any other options. Not long term. Strict obligate anaerobes, on the other hand, require the absence of oxygen. Why? Because, one, they don't use it, but why they require to not have it is it will do what? It would kill them. Right? It would kill them. Microaerophiles, micro means what? Small. These ones require small amounts, less than atmospheric conditions, which is what exists in this room in most places. If we were in a microaerophile type environment, you would pass out. There is a subset of microaerophiles known as capnophiles. And these guys not only don't really need oxygen at high levels, they need it at lower levels, but they need higher than normal levels of carbon dioxide. So this is where a candle jar would be beneficial to them, because when you burn a candle, what gases are produced? Carbon dioxide, and then the bad one for us, although carbon dioxide is bad too in large quantities, carbon monoxide.
When you're at high elevations and there's less oxygen,
Mm-hmm.
what is it that's taking the place of there being less oxygen, or is it just that the air is thinner, so there's just less?
Yeah. I don't know why it is less oxygen, but there is.
Right.
At higher elevations, there is less oxygen. I would say, I guess, because it's further away from the source that's producing it. Because think about it, the major overproducers are in our oceans, which are at sea level, because they're ocean. And so that when you get the higher, the concentration is less. You're further away from the source that's producing it. That would be my best educated guess on that one. But it's not good for us, right? Because we're not used to that. And so, the body doesn't do so well. These guys have got it made, facultative anaerobes, because as their name implies, they can grow anaerobically. They do not require oxygen to grow. But they can grow without it. I mean, they don't require it to grow, so they can grow without it. But they can grow when it is present, and they can grow really fast, because you get the best amount of energy per molecules you break down using oxygen as your final electron acceptor, as opposed to any other electron acceptor or any fermentation process. So most of the ones that we work with in this lab are facultative, and you guys will find that out soon. The last, I don't have any more of these because the one I had was a pathogen, so I had to get rid of it. I got to find us another one. I don't have any aerotolerant anaerobes. And so these guys don't use oxygen for their metabolism, but they can grow in the presence of it. They can tolerate, as their name implies, aerotolerant. So as it relates with some of these definitions, it's good to look at, do they require oxygen for their growth? Can they grow in the presence of it? And can they grow without it? Right? So I'm not going to go through this one. I want you guys to try and fill this out for yourself for homework. If you're in my lecture class, we've already done this, right? We did it pretty quickly, but it was recently, right? So try and fill this out, and then if you download the PowerPoint right and you click, it tells you the answers, okay? So for this media, you'll notice in the demo tubes that I have for you guys today, there's a little bit of agar, not as much, of course, that's in our motility. But there's a little bit of agar in this tube, and because of that, for the demo tube that I have for you guys today, you can see that growth suspended in that tube. The reason why that agar is in there is to slow the diffusion of oxygen into those tubes, right? These aren't screw cap tubes, are they? They are loose cap tubes. They allow oxygen to enter in. So over time, and that's why your guys are, he already got me, Peter, something else?
I was just going to put a couple my class media.
Uh-huh.
That fruit's not labeled.
Okay, it's yours, okay, yeah. I won't throw it out.
Sorry. You remember asking me for a micro book a
Uh-huh.
while ago, and it wasn't in my office?
Uh-huh.
I found two at home. What was the name?
Wesner.
I'll have to look and see. Do you still need it?
I'm good, but thank you. I'm glad to know that I did give it to you, though. Hopefully. Okay, so it's going to slow the diffusion of oxygen into this tube, because remember, the goal we want an anaerobic environment in the bottom of this tube. So one of the things I had to do, though, because these are not closed cap tubes, is that morning I came in and I boiled the oxygen out of the tubes, right? So I forced the oxygen out, and then it slowly diffused back in. So remember you had this little pink layer in your tube? It also helps stabilize the growth, so you can kind of see a growth pattern in the tube. So we'll look at that tube in a moment. The other components of this media are sodium thioglycollate and L-leucine. What do these do? What do these do? Right, they take that molecular oxygen, that gaseous oxygen, and they turn it into water. So again, we want to create an anaerobic environment in this tube. So that's what those reagents do, is actually convert the oxygen into water. What color is the resorant in the presence of oxygen? So that's an indicator that's in this tube. It's pink, because can you see oxygen with your eyes? I can't see oxygen with my eyes. I can definitely feel it though, right? Because we would pass out, like I said, if there wasn't any oxygen. But you can't see it. So we need something to help us visualize, right? And so I'll pull out some old tubes so you guys can see, and the whole tube is pink, right? Because the oxygen diffused all the way down into that tube. Would you want to use those tubes? Not if you're trying to figure out if your organism can grow anaerobically, because is that tube anaerobic? No, the whole thing is pink. That means it's full of oxygen. Right? You're not going to be able to get the answer to your question. So this gets oxidized, right? It gains electrons. No, loses. Oxidized is the loss of electrons. Oil rig, right? Yeah, oil rig. Oxidation is the loss of electrons. Reduction is the gain. So it will lose electrons, the indicator, to oxygen, and turn pink. So why do we want that band to be thin as opposed to being really thick? Right, we don't want a lot of oxygen because this is where the highest concentration of oxygen is. That doesn't mean there isn't some oxygen right here below this pink layer, right? There's a little bit. So if this is really thick, then the little bit of oxygen, I mean, you're left with like this much anaerobic zone, right? Little tiny section at the bottom of your tube. So how are you going to tell if your organism can grow anaerobically if you left him only this much room? It's going to make it hard, right? Definitely going to make it hard. So based on the growth pattern in these tubes, right, if you use fresh tubes, right, that have a thin band, you inoculate it properly where you inoculate the whole tube, right, this is the one where we inoculated from bottom to the top, then if your organism is an obligate aerobe, where should you see growth in these tubes? Which tube represents the pattern you should see? Number 1, right? You should only see growth in this oxygen-only zone. So some of your tubes, right, I put them in the refrigerator so they would slow down growing, right? Because if I left them out, the oxygen keeps diffusing in, diffuses all the way to the bottom, you see growth through the whole tube. So I put them in the fridge to slow them down, right? And also so you're not jostling them around, but so that you'll see at the top of your tube is the only place where you have growth. So there's a few of you guys, that's the case. You have an obligate aerobe. It has to have oxygen to grow. If you had a microaerophile, which we don't have in this lab, because again, there's too much oxygen in this lab, where would their growth pattern appear? Which one of these tubes? It would be number 4, and that's, again, remember the greatest concentration is at the top, right, where oxygen is diffusing in, and then below that, right, or just at the bottom of that oxygen zone where there's less oxygen is where these guys would grow. Again, nobody has one of these. Don't write it. Okay. Obligate anaerobes, where would you see their growth? 2. Only at the bottom of the tube, right? Does anybody have one of those in this lab? No. Oxygen would kill it. Aerotolerant anaerobes, where would you see their growth? 5. But 3 and 5 look very similar, huh? What's different between 3 and 5? Right, the concentration at the top of the 3 is greater, which means oxygen does affect their growth, right? It actually allows them to grow a lot faster than this guy where it's uniform growth in the oxygen and non-oxygen zones. That one's your aerotolerant because it's tolerating oxygen, right? It's not killing it, but it's not using it. It's an anaerobe. Most of you guys have this one, which is a facultative anaerobe, where if you're lucky, you will actually see increased growth at the top of the tube. But you will see growth throughout your entire tube, and you're supposed to, because yours can grow anaerobically. Mostly it ferments. Okay? So if you look in your racks on the side that just has 2 tubes labeled A and B with white stickers, you'll see those 2 examples. You'll see the facultative anaerobe and the obligate aerobe. So which one is the obligate aerobe? B. Do you see how it's just at the top? And then the other one, that organism, you can really see the much thicker layer, and you can even see the stab that I did through the anaerobic layer. That's a really good example of a facultative anaerobe tube A. So go ahead and make sure you take a look at those, and we'll compare them. So here's some actual tubes, right? Instead of little dot plots. So our strict aerobes, remember, require oxygen, only grow in the oxygen zone. So which one of these tubes, A, B, C, D, or E, do you see growth, cloudiness, only at the top of the tube in the oxygen zone? D. What about your strict anaerobe? This one's kind of tricky. So what part of the tube is anaerobic? Anaerobic. Aerobic is where the oxygen is, right? This pink layer. So on this tube you can compare across, right? So what is this region? This is anaerobic, right? So you don't see any growth in this tube in that anaerobic zone. You don't see any growth in this tube in the anaerobic zone. We already said this is your aerobe, right? This one you see growth in the aerobic zone, and look, you can see the pink layer. There's no growth, right? That one is your strict anaerobe. This one, look, you've got lots of growth in the aerobic zone, little bit in the anaerobic. This is the facultative. Where's our microaerophile? E. See how it's right there at the bottom of the oxygen zone, right under the pink? Or the very bottom of the pink. So there's our facultative. So that leaves us, see how it's uniform growth throughout? That's your aerotolerant. And some of yours kind of look like this, but trust me, it's not. It's a facultative.
So if the reason why like E still or D doesn't have any peaks is because the organisms used up all the oxygen that was available.
Yeah, and its growth is clouding it out, so you don't see it. So you don't see that pink layer. So she was asking a good question. So both B, and even this one, you don't really see the pink layer, and that's because the growth is obscuring it. Okay, so why is it important this media be fresh? We want it to be what? We want it to be anaerobic. If it's not anaerobic, then we can't test for anaerobic growth, right? So you got to use fresh media, or you got to boil out the oxygen like I did. Scientific method. Okay, so for the anaerobic jar, although this one's starting to lose its color because it's old, I need to take out a new one. The methylene blue strip, because again, we've got to use an indicator. We can't tell that there's oxygen in here, right? We need something to help us. Methylene blue, which is a dye we use, right? What color is it under aerobic conditions like in this lab? It's blue, but when you stick it in anaerobic conditions, it loses its color. It becomes colorless. This one's becoming colorless just because it's old. I need to open a new pack. Maybe I'll do that. So I got packs. Just don't have jars that will seal. There it is. It's like, I know there's a tear here somewhere. Right? So there it is, because this was sealed up in an anaerobic situation. When you open it up and you put it in this aerobic situation, this was anaerobic without oxygen. When I take it out of that packet, it's aerobic. It'll start a chemical reaction, and so this packet in this jar would start releasing two gases. So methylene blue under aerobic, right, is blue, but under anaerobic it is white or colorless. Ooh, I can feel the reaction. You can feel it. Pass it around. It's getting warm. Yeah. So there's one set up in the jar. You see how it's clear? So I knew it was working, right? That jar became anaerobic. The reason why is there's a chemical reaction happening right now with that packet. It's releasing two gases. So that jar, when I took the picture, was under anaerobic conditions, right? Because that tablet isn't blue. Here's one I left out. You see how it's blue? And that's what's going to happen to that one if I leave it out. That tablet's going to turn blue. Anyone want to warm their hands some more? So it's releasing hydrogen and carbon dioxide gas. The hydrogen gas combines with the oxygen gas, and it forms, yet again, water. So that's how we remove the oxygen out of this jar, right, is this chemical reaction packet. It's going to take a little while, but it will. So this, why do we see the condensation when I put them in the incubator? Because we're making water. So it actually will even condense on the outside or the inside of the jar, on the walls. So there's one instructor that's not here anymore. The size of this jar determines how many packets you have to put in, right? For this jar, you'll notice that it holds 3 packets in its little tray in here. One teacher, she's like, oh, I only put, like, you know, half the plates in there, so I only put 1 or 2 of these in there. I'm like, well, that's not going to work. You got to start all over. She's like, what? Why? I'm like, it's not about how many plates you put in the jar. It's about how big the jar is and how much gas you got to produce to get rid of the oxygen to form the water. No, she just doesn't understand she didn't follow. She didn't understand the instructions.
Okay. I thought this was something that's like instilled in your mind when you were
No, I mean, when you learn something new, right, you're given instructions. She wasn't either given good instructions or didn't understand them and was trying to save on the gas packets, but instead that made it not work. Okay, so if the environment in this anaerobic jar, right, is actually anaerobic, we put enough packets in the jar, right, to make it anaerobic, how would that affect the growth of an anaerobe? So if we stuck an anaerobe in here and we suck the oxygen out, we just made this guy what? Happy. He's going to grow. If you stick a known aerobe in this jar and you suck the air out, what are you going to do to him? You're going to kill him. What about a facultative anaerobe? He's going to grow a bit slower, right? He's going to go, I got this covered. I'll just kind of poke along. Slow as it may be, but I'm still alive. You can't kill me. When we do this experiment, we do a set of plates in the jar and a set outside the jar, which of course is aerobic conditions, right? So we can compare. So an anaerobe, what's going to happen if we just leave it outside the jar? It's going to die, right? An aerobe, on the other hand, is going to go, I've been rescued, right? I can breathe, right? It's going to grow. What about that facultative anaerobe? How's he going to feel about that oxygen? He's going to grow, and he's going to grow like crazy, right? Because oxygen's the best, right? It's going to give you the most energy. So if you plated a certain number of bacteria, you could probably even see those different growth patterns, right, between inside and outside for a facultative. Where with your aerobes and anaerobes, it's going to be grow or not grow, right? So If it's colorless, right, then we know the environment inside the jar is what? If we have a colorless, it's anaerobic. It's already starting to turn blue. It's hot though, man. I'll steal some of these for later when it gets cold. Mardi Gras time, right? Okay, so no oxygen, right? Outside the jar, we know, right, in this lab is what type of environment? Aerobic, and there is definitely oxygen. All right, so for Pseudomonas aeruginosa, this organism, when we put it in the jar, there was no growth. Outside the jar, it grew. Clostridium sporogenes grew inside the jar, but not outside. Staph aureus grew inside the jar, but grew better outside the jar. So how would we classify these? What would Pseudomonas be, since it only grew under aerobic conditions? It'd be aerobic. And since Clostridium sporogenes only grew under anaerobic conditions, it's a anaerobe. This guy grew both conditions grew, but grew better under aerobic, so it is a facultative anaerobe. Again, I'm going to skip over the rest of these. I want you guys to go over them yourselves. So, we already kind of answered this one, right? Who would really like the candle jar? Capnophiles, because when you burn a candle, you release carbon dioxide gas. Right?
So all capnophiles are microaerosols.
Yes. Small amounts of oxygen, but in addition, they need lots of carbon dioxide. All right, so let's get to what we need to do.